Hello, hi, Assalamualaikum dan selamat sejahtera to all my lovely girls in 5 best tarif, 5 cerdas and also 5 dynamic. Okay, so let's continue our last subtopic in the chapter 13 with the 13.2 biotechnology. Alright, okay, so let's go to the learning standard that you have to cover for today. Okay, so number one, explain the meaning of biotechnology. Okay, number two, describe applications of biotechnology in life. And the last one, justify the importance of biotechnology in life. Okay, so these are referred to the learning standard that you have to cover for today. Okay, so let's go to the biotechnology definition. Okay, what is a biotechnology? So biotechnology is a field which utilize technology or methods to manipulate organisms for the production of biological products. For the production of biological products. Okay, that is referred to the meaning of biotechnology. Okay, I will repeat. So biotechnology is a field which utilizes technology or method to manipulate organisms for the production of biological products. Example of the biological products are food, and then also beside foods, animals, and also refer to the plants. Alright, okay. Now we go to the aims of the biotechnology. Okay, what is the purpose of the biotechnology? Okay, biotechnology aims to improve the quality of livestock, and crops, untuk mempertingkatkan kualiti haiwan ternakan dan juga tanaman, hasil tanaman, as well as to develop the application of microorganisms for specific purpose. Okay, that is referred to the purpose of the biotechnology, to improve the quality of livestock and crops, right? As well as to develop the application of microorganisms. Okay, right. Let's go to the still to the biotechnology field. So since biotechnology involves various disciplines of science, so therefore each discipline that involves the use of living organism for a specific purpose is given a specific color code. Is given a specific color code uh, based on the disciplines of science. Okay, so although 10 biotechnology activities are identified, okay, so there are 10 biotechnology activities in this field. So the main ones of the biotechnology activities are green, white, yellow, and blue biotechnology activities. Okay, let's take a look on the biotechnology activity based on the various discipline of science. Okay, so this is referred to the types of biotechnology activities based on colors. Alright, okay, so there are 10 biotechnology activities, but the main one is referred to the color code that has green, white, blue, and yellow. Okay, so this is referred to the main biotechnology activities, right? Okay, so let's go to the application of biotechnology in life. Okay, so why the biotechnology is very important? Okay, later we will discuss about that. Now we are going to discuss the application of biotechnology. okay, in our life. Okay, first in gene therapy. All right, gene therapy. So what is gene therapy? Later we will discuss about that. Okay, and then also in the DNA profiling, production of insect-tolerant plants, and last one is cleaning of oil spills. Right? So the application of biotechnology in life involve the gene therapy, DNA profiling, production of insect-tolerant plants, and also for cleaning of oil spills. Okay, so these are referred to the application of biotechnology. Alright, so let's go to the first application of biotechnology that involved in the gene therapy. Alright, so what is the gene therapy? So gene therapy is used to treat or prevent, to treat or prevent genetic disease. Okay, do you still remember what are the genetic disease that we already discussed in the chapter 11, in the chapter 12. Okay, so examples of the genetic disease such as hemophilia, yeah, thalassemia, sickle cell anemia, also all of them are genetic disease. So gene therapy is used to treat or prevent the genetic disease. So in gene therapy, a normal gene is inserted into the patient to replace the abnormal gene, right? So the abnormal gene will be removed and a normal gene will be inserted into the patient. So the disease can be cured. Yeah, the disease can be cured by gene therapy. So example of the disease that can be cured by gene therapy are cystic fibrosis, okay, refer to the damage of the lungs, the excessive mucus of the lung that cause damage to the lung. So by gene therapy, it will cure, the cystic fibrosis, and also muscular dystrophy, the degeneration of the muscle cells, and a few types of cancer. Okay, so that is the use of the gene therapy. So gene therapy can cure the certain disease. Okay, so these are referred to the basic steps in gene therapy. Yeah, the basic steps in gene therapy. Okay, first, a cell with an abnormal abnormal gene, right? Okay, so this cell contain gene that is abnormal. Yeah, so this is referred to the abnormal gene. Right? Okay, number two, an insertion of normal gene to replace the abnormal gene using a virus. Okay, so the abnormal gene will be removed. And will be inserted with the normal gene. Okay, by using a virus. Okay, and then last one, the cell perform normal functions. Because now the gene is normal, no more abnormal gene. So the cell can perform the normal functions without the disease. Okay, next we go to the DNA profiling. The DNA profiling. Okay, so what is a DNA profiling? DNA profiling is a technique used in forensic, to identify individuals, based on their DNA, based on their DNA. Okay, so it is a very important technique in the forensic, because sometimes we want to identify the criminals, so we use their DNA to identify the individuals. So this is due to the fact that an individual's DNA is unique. Yeah. So every individual in this world has different DNA. Yeah, has different DNA. So that's why DNA is unique. Yeah, except between identical twins. So the identical twins will have the same DNA. Yeah, but the others have different DNA. A human DNA set is different from animals. Yeah, so of course, human DNA is different from animals. We have a different number of chromosomes between human and animals. Alright, so an individual can be identified based on the DNA obtained. So the DNA will be obtained from his or her blood. From blood, semen, for the male, or the skin. Alright, so most of the DNA is obtained from the blood, semen, semen just for male, semen ini maksudnya air mani, bagi lelaki, or skin, right? Okay, skin. And then the DNA profiling technique was invented by Sir Alex Jeffreys in Leicester University in 1985. Okay, so this is referred to the inventor of the DNA profiling. He was awarded for his achievement the Copley Medal, the oldest award before the Nobel Prize was introduced. Okay, it is about the DNA profiling. Okay, so besides the blood, semen, or skin, DNA also is obtained from the hair. Right, okay, the application of the DNA profiling. Application of DNA profiling. So, to identify criminal. Untuk mengenal pasti penjenayah. So, to identify the criminal. And to settle the paternity and maternity dispute. Untuk menyelesaikan masalah ibu, the biological father, the biological mother. Alright. Okay. And then to identify the genetic disease. Identify the genetic disease such as cystic fibrosis, thalassemia, sickle cell anemia, hemophilia, and so on. Okay. Determine the suitability of organ donor and recipient. Okay. Determine the suitability of organ donor. So DNA profiling is very important in organ donation. In organ donation, to determine whether it's compatible or not. With within donor and also the recipient. Okay, so these are referred to the application of the DNA. Make sure you can memorize it, right? Okay, we go to the steps used in DNA profiling. Okay, so what are the steps? The steps that have been used in the DNA profiling. Okay, first, the blood, semen, or skin sample is obtained from an investigation scene. For example, a crime scene. And then the DNA is extracted from the sample. Okay, DNA is extracted from the such as blood, skin, from the semen, from the hair. Alright, okay, and then restriction enzymes cut the DNA into different size of DNA fragments. Okay, now DNA fragments are cut. Are cut into different size. Okay, by using the restriction enzymes. Okay, then the DNA fragments of different size are then separated by gel electrophoresis. Okay, so now the DNA fragments are separated into different size. Okay, by using what? By using gel electrophoresis. Okay, then DNA fragments of different size are transferred from the gel to a nylon membrane. Okay, then the radioactive probes are added to the nylon membrane. Okay, X-ray film is then placed on top of the nylon membrane, and the X-ray film is possessed to show the position of DNA bands that form from the DNA profile. Okay, so at last of the process, okay, it will show the position of the bands, position of the DNA bands, of the person. Alright, okay. So by using the X-ray film. Okay. So these are referred to the example of the X-ray film, X-ray film that show the position of the DNA bands, DNA bands, of the different DNA profile. Okay, so please read the question here. So based on the DNA profile below, can you determine who is the biological father of the child? Okay, so we want to determine the biological father of the child. So this is referred to the DNA profile, DNA bands of the mother. So this is referred to the DNA bands of the child. And then, okay, so these are referred to the possible father of the child. Okay, father 1 and father 2. So, to determine the biological father of the child, okay, we have to look at the DNA bands. Okay, so which of the DNA bands of the father 1 and father 2 is closely or same with the child? Which band of the father 1 and father 2 which has the same position, the same position of the band with the child. Okay, let's take a look on the bands here. Okay, so this is father 1 and father 2. So this is a child. So all of them have the same position for the band 1. Okay, we go to the band 2. Okay, band 2 and then band 3. Okay, so band 3 here for the child. For the child. So the father 1 has the same position of the DNA bands. DNA bands. Okay, this one also. The father 1 also have same position of the DNA bands. Okay, so maybe the father 1 is the possible father. Okay, so let's take a look more on the next band. Okay, so this one. Alright, so this is referred to the DNA bands, for father 1. Alright, and this one. Alright, okay. So, alright, okay, because of the father 1 has more, right? Has more, has more position of the DNA bands that's same with the child. So father 1 is considered or Alright, so is the father, is the father of the child. Alright, so the father 1 is the father for the child because most of the bands of the DNA and the bands of the DNA of the father 1 and the child are the same. Are the same. But for the father 2, I think no, just a little bit, maybe one, so no, they have no same position of the DNA bands, in the father 2, but father 1 have about 4 same position of the DNA bands. So the father 1 is the father for the child. Okay, so that's how they use the DNA profiling to determine the biological father or the biological mother of the child. Okay, now we go to the production of insect-tolerant plants. Insect-tolerant plants. Okay, so farming cotton breed. Farming cotton breed with high yield and resistance to pests such as BT cotton. Right, BT cotton. Okay, in cotton farms has reduced the use of pesticide. Okay, so what is a BT cotton? Later we will discuss about that. Okay, so the BT crops. Okay, what is a BT crop? BT crop refer to the crop that is injected with genes from Bacillus thuringiensis or BT. Right, so BT crop refer to a crop that have been injected with the genes from Bacillus thuringiensis, okay, a type of soil bacteria that secrete toxin. Now, so when the BT crop, when the crop is injected with the gene, from the BT or from the Bacillus thuringiensis, now the crop can produce the toxin. Yeah, the crops can produce the toxin. So when the crops produce the toxin, now the crops is tolerant to the pests, yeah, or to the insect. Yeah, okay. So this effort has saved costs by reducing the use of pesticides and also increase cotton yield. Right? Okay. So that is referred to the application of the biotechnology, whereby the gene of the Bacillus thuringiensis, which is a type of soil bacteria, that secrete toxin. So the gene from the BT is inserted into the crops, into the gene of the crops, and will cause the crops to produce the toxin. So when the crops produce the toxin, it will be tolerate to the insect, tolerate to the insects, and cause it will cause the insects die. All right, okay. So this effort is very important to save the cost of the use of the pesticide, and also in the meantime it will increase the cotton yield. Okay, beside cotton, other BT crops are corn and brinjal. Alright, so these are referred to the BT crops, corn and brinjal. Okay, so let's go to the diagram here, comparison between normal crop and GMO crop. Okay, so GMO crop here, what is a GMO? Genetically modified organism crop. Okay, so why we call as GMO crop? Because now this crop have been inserted. The gene of this crop have been inserted with the BT. BT gene. So what is a BT gene? Just now, BT refer to the Bacillus thuringiensis. Okay, so this is referred to the normal crop of the normal cotton, cotton here, and here refer to the BT cotton. Okay, so you can see the difference between the normal crop and between the GMO crop here. And here refer to the normal corn, and this is referred to the BT corn. Normal corn and BT corn. All right. So what happened to the BT crops? Okay, so genes that produce BT toxin from Bacillus thuringiensis is inserted into the plant's genome or the plant's gene. Okay, so the gene from the Bacillus thuringiensis, okay, that produces a toxin, that produces a BT toxin is inserted into the plant's genome. And then, okay, when the pest feeds on the plants, when the pest feeds on the plants, which produce a BT toxin, so what happened to the pest? The toxin affect the digestive system of the pest and kills it. Alright, so now the toxin that produced by the plants, will kill the pest. Alright. Okay, so that is referred to the effect of the BT crops on pests, whereby the BT crops will produce the toxins, and eaten by the pests, and it will kill the pests. Right. Okay, so that is referred to the effect of BT crops. Okay, next we go to the cleaning of oil spills. So oil spills sometimes happen, in the sea, right? Okay, so bioremediation is a method which uses bacteria to clean up the environmental pollutants. So bioremediation. Bioremediation. It is a method that uses bacteria. To clean up the environmental pollutants. So it is effective to treat oil spillage in the ocean. Effective to treat the oil spillage. Most molecules in crude oil and purified oil products can be degraded by bacteria. Okay, so for example, Alcarnivorex borcumensis. Okay, it's a type of the bacteria, yeah? Type of the bacteria that depends on oil for its source of energy and is widely used to treat oil spills. Okay, so in the in the bioremediation, we use the bacteria to clean the oil spills in the sea. To clean the oil spills in the sea. So what happened? The bacteria will produce the bacteria will break down the oil. Will break down the oil to produce the energy, and then it will cause it will treat the oil spills. It will clean the oil spill.